Hi, and welcome to Mr. Fisher's Biology Podcasts. This podcast is on DNA replication. So what is DNA replication? Well, we know that cells come from other cells, and in order for our body cells to be identical in terms of their DNA, new cells will need identical DNA strands. And so DNA has to be copied before a cell divides. And during interphase, there's a stage called the S, or the synthesis phase, where this takes place. So the synthesis phase is located in the cell cycle during what we call interphase. And this occurs in the nucleus of eukaryotes. So this is our cell cycle, and this right here would be the M, or the mitosis phase. So this is where the cell divides. The synthesis phase is located sort of midway through, and that's the S phase. This is where DNA replication occurs. So DNA replication begins at areas known as the origins of replication. And in the origins of replication, the two DNA strands, because remember it's a double helix, start to open up and form replication forks. So right there, that's a replication fork. Now new strands will begin to grow at those forks. So DNA replication, as the two DNA strands open up, we get replication bubbles. And a replication bubble is different if we're a prokaryote. So prokaryotes have a single bubble, and eukaryotes have many. So this example here would be a eukaryotic chromosome because there are many replication bubbles. And that's a replication bubble there. So beginning in DNA replication, it starts with the enzyme helicase. And helicase will begin to unwind the DNA strand, and it does that by breaking the weak hydrogen bonds. In this image here, helicase is depicted here. And helicase will begin to move into that DNA strand, break the hydrogen bonds, and separate the two strands. Now at the same time, we have single strand binding proteins which attach along the DNA strand. We have some single strand binding proteins there, and that helps to keep the strands separated and untwisted. Now, on top of that, the enzyme topoisomerase will attach to the two forks of the bubble, and it relieves the stress on the DNA molecule. So this is topoisomerase right here, and topoisomerase simply starts to unwind the strand early. You can think of it like a rope, and that prevents stress from being placed on the DNA molecule. Now, before the new strand can form, there must be RNA primers present, and primase is an enzyme that synthesizes an RNA primer. So RNA is actually added to a DNA strand prior to replication, and this sort of primes the DNA strand, which is ready for this guy here, DNA polymerase, will add the new nucleotides. So looking at this is DNA polymerase here. What we don't see are the RNA primers. We do see them up here. Those are RNA primers. And there's little chunks of RNA that signal to DNA polymerase that it's time to add on. Now, an important point to note, DNA polymerase can only add nucleotides to the 3' end of DNA. So notice we have these here. What these constitute, or what these signify, are the types or the numbers of sugars that are found on a deoxyribose. So we label, sorry, not the sugars, but the carbons. We label the carbons by numbers. And so DNA polymerase can only add nucleotides to the 3' or the number 3 carbon on a deoxyribose. So what does this all mean? Well, this means that the new strand has to be built in a 5-3 direction. So if DNA replication, or if the fork is opening here, then the 5' end, we can follow it, DNA replication is occurring in the 5-3, right? And notice that we have 3 to 5 on the opposite side. Same thing up on the top strand here, which we'll get to. This is known as our lagging strand, but it's still being built in a 5-3 direction. Okay, so what this means is we have two strands, as the two strands are forming, one, which is called the leading strand, that's this strand here, is synthesized as a single strand. So the direction of replication on the leading strand is the same as the direction of replication from the point of origin, where the DNA strand is opening. In other words, DNA polymerase can slowly move along and add on new nucleotides. That's our leading strand, still in a 5 to 3 direction. The lagging strand synthesizes discontinuously. So if we look at the top, which is our lagging strand, as the replication fork opens this way, notice that the DNA replication on the lagging strand is backwards. So in other words, there's a lag in which nucleotides can be added. DNA polymerase needs to wait until this opens up more, and then we can synthesize more. So in other words, what we end up with are very short segments. Now, these segments are called Okazaki fragments. So this is an Okazaki fragment right here. And Okazaki fragments must be joined by an enzyme at the end of replication. And that enzyme is ligase. So DNA ligase will join those Okazaki fragments to make one complete strand. Now, when we look at proofreading, DNA polymerase makes about 1 in 10,000 base pairs errors. And remember, errors are mutations, and errors can be fatal. So we need to proofread the DNA strand. And enzymes will proofread and will correct those mistakes. After the corrections have taken place, the new error rate for DNA is about 1 in a billion base pairs. So notice how much different the error rate is. Now let's look at the semi-conservative model of DNA replication. This model was proposed by Watson and Crick. And what it suggests and what it sort of signifies is that the two strands of parental DNA act as a template for the complementaries. So if this is my original parent strand, notice that after DNA replication takes place, that I have a parent strand on this side, I have a parent strand on this side, and they act as templates for the complementary strands. So DNA is composed of one parental strand and one new strand. And in this way, it keeps the code fairly intact. Now, some little bit on DNA damage and repair. Different types of chemicals, as well as ultraviolet radiation, can cause damage to DNA. And so cells must continuously repair damaged DNA. Anytime DNA is damaged, what's called an excision repair occurs, where a piece of DNA is removed and then enzymes, DNA polymerase and DNA ligase, can replace and bond the new nucleotides together. So DNA is constantly being repaired, it's constantly being corrected, and in this way it keeps our bodies fairly safe. So let's finish with a question, and this will conclude the podcast. So what would be the complementary DNA strand for the following DNA sequence? Try it out, and I hope that was helpful.